Hi everyone, you're welcome. So we're going to be having a quick walkthrough on the specimen. This is not a repeat of the analysis on the specimen, but this is an exam style kind of questions that will prepare you for the exam as you walk in. We will not make this very long. We'll just quickly see how we can walk page by page to see the answers in case you're asked to put up something like that in the exam. So at any point it is too fast for you, you can pause the video, write down the elements, and we move as we continue. So let's go to the first question. Study specimen A and B and identify the questions below. So you're asked to identify specimen A and B, and you're asked to classify them agriculturally by seed, leaf, and by life cycle. You are asked to state the type of fruits you have in specimen A, where specimen A and B is gotten from, and then you're asked to state the reason for the type of fruits you have just mentioned. You are asked to also mention the two agents of dispersal for specimen A and B. And then here you're asked to mention three diseases of the crop from which specimen A is obtained. So having that in mind, let's quickly see a flash of the answers. So basically here you see that specimen A, we identified it there, and specimen B, and then we classified it agriculturally. We classified it based on the seed leaf and then by the life cycle as well. And then we say the type of fruit is a legume, and the reason is because it's a dry dehiscent fruit that's the pericarp splits when it is dry to release the seeds. Now we mentioned the agents of dispersal, and then we mentioned the diseases that you can get from specimen A. So of the crop from which specimen A is obtained. So you have the leaf spot disease, you have the rosette disease, and you have the root rot disease. Moving on to the next page, we say here that you are asked to state the type of germination in the plant where specimen A is derived from with a reason, and then you're asked to mention three biological importance of specimen A to the soil. And here you're asked a very interesting exercise here to rub specimen B on a white paper, record the observation and the result in a tabular form. So expect something like this in your exams. It's going to come, I'm very sure this is going to come in different ways. So you have this now, we're going to look at that, and then you're asked to mention three reasons why the crop of specimen A is best cultivated in specimen E. So let's just look at a flash of the answers as we move on. So for the answers here, you'd see that the type of germination in specimen A is your epigeal germination, cotyledon above the ground. Three biological importance, you have them here. And then let's go to the test and lay some emphasis there. So you have the test here. The test, you must always tell them what you did. So small portion of B rubbed on a white towel. The observation, you have a translucent oil spot allowing light to pass through, and then the inference is that your lipid or your fat and oil is what is present. This carries most times 6 marks, so you have to take note of that and be very careful with how you put down your answers. The next one says mention three reasons why the crop of specimen A is best cultivated in specimen E. So you can pause and write that down as well. That's going to be very helpful to you as you go into the exams. So let's move to the next one. I told you this video is going to be very short video, so we are trying to be very fast and working with time. So the next one is complete the following table on specimen F. Okay, so specimen F, you have to classify it into the kingdom, the phylum, and the class. So let's quickly see all the questions here before you look at the flash. Now mention three adaptive features of specimen F. We are asked to do that as well. Remember adaptive features, we talked about it earlier, that you must mention the parts and the function of that part. The next one is states the feeding habits and possibly type of food in organism with specimen F. Okay, so you have to take note of that as well. And then the next one is that you are asked to state the ecological relationship between specimen G and its host, giving a possible reason for your answer. So let's see the flash, the answer flash as we move on. So coming here now. All right, so look at the answer flash now. So we've stated that this belongs to specimen F belongs to Animalia, Chordata, and Mammalia. And then we give the adaptive features. We talked about this. We are not emphasizing this again. Just go back and read that up for yourself. And then we're talking about the feeding habits. So herbivorous, it can be herbivorous and then they feed on herbs. It can be carnivorous, they feed on flesh. And then when they are omnivorous, omnivores feed on both flesh and herbs. So please take note of that. That's very, very important, depending on the one you wrote down there. And then the ecological relationship between G and its host, we said it's semi-parasitic or hemi-parasitic, and this is because it has a green part for photosynthesis as well as taking nutrients and water from the host. Okay, so that's that for that particular set of answer. Like I said, if it is too fast, pause the video, put some things down as you move on to the next one. All right, so let's go to the next page. The next page says study specimen H carefully and use it to complete the experiment. This is also very important. Please pay attention to this. This is also very, very very, very important. So they said add a small portion of specimen H to a beaker and then add drops of sodium hydroxide. Take note of this. And then you are asked to also add elements of copper sulfate, copper 2 sulfates to this mixture. So you're going to have this, and this is going to be a whole lot of 6 marks. So please take note of this question. You can't afford to lose out careless marks. That's what I tell my learners and my students. You don't lose marks that you're not supposed to lose. Don't lose careless marks, okay? Make sure that every point that, let's say that you lost a mark for a question you deservingly, okay, are supposed to lose the mark, okay? So please take note of that. Now let's go to the next one. Here you're asked to complete the table on specimen J. Here, kingdom, phylum, and class, and then mention 3 adaptive features again of specimen J. So we are also going to look at the answer flash to see the possible answers for that. So looking at the answer flash, you see here that what you had here was that after adding a small portion of H to sodium, or adding sodium hydroxide to your H and then adding your copper 2 sulfate, you had the mixture turning purple. That's your observation. What you see actually is your observation, and then the inference is that protein is present, and this test is called the Biology test. So you want to write that down in case you're asked. The name of this test is a Biology test, okay? Take note of that. Moving on to the next one, they asked us to complete the specimen, the classification of specimen J into kingdom, phylum, and class. So you have Animalia, you have Chordata, and you have Pisces. So please take note of that. This is very important. And of course, you all know this. We don't want to talk about this again. Adaptive features of what? Specimen J. Fin, gills, scales, and streamline bodies, together with the functions of what they do. So please take note of that. That is very, very important. So moving on to the next question. We are just trying to see how we can make this very fast. Now the next one says complete the table on any 2 classes of food provided by specimen J and their functions. So you see that expect this kind of questions as well. Classes of food and 2 functions of each. So you see, you could have your protein here and you could have your fat and oil here, and you must know at least 2 functions of each of these classes of food. And then here you're asked to make a diagram. Of course, you know we can't say less of a diagram. You will definitely be asked to draw. We don't just know what you'll be asked to draw, but you will definitely be asked. So practice over and over and over and over again. The fear of practicing keeps you away from drawing, but by the time you draw the first and second and the third, you get used to the diagram and your confidence level will go very high as you prepare for the exams. Okay, so let's look at the answer flash as we move on. So the answer flash for that group is that here you have proteins. Okay, so proteins help to do what? They build and repair the tissue and they support the growth and development. And fat and oil, of course, provides energy and insulates the body. So take note of that. Pause the video and write it down. So for the diagram, remember we talked about diagrams in a detailed way in the previous video, but to re-emphasize the importance of a diagram, because you see now that in this question I had to assign a whopping 8 marks to the diagram. So you need to have your title, you need to have your size, you need to have your clarity of lines, neatness of labeling, your details, and then you have your words, your magnification. This is very important. So the title, I'll mark myself correct. Side view of specimen J, I got that. And then my size, if they said 8 cm to 10 cm, yes, this is most likely going to be 8 cm to 10 cm. So I would also give myself a mark there. That's how to share the marks. Clarity of lines, please don't use broken lines. Use continuous lines. So in this case now, if this were to be a pencil sketch, it would be I would give myself a mark because the lines were continuous. So I would give myself a 1 mark there. Neatness of labeling, you notice that I always advise that you label to the sides. in this case I couldn't just label to one side, so I had to label to both sides, but I labeled horizontally. So I think I got my mark. I deservedly got my marks for my what, for my neatness of labeling. So I'll give myself a 2 mark for my neatness of labeling. And then my details as well. This looks like a fish. Of course, I can see my dorsal fin, I can see my eye, I can see the caudal fin, and all of that. So I'll give myself maybe 2 for details again, and my magnification was well stated here. Okay, so you have to also do that for yourself. And then remember, when you write your magnification, you must always put a times in front. I put times 2 in front, so I get my mark. So maybe I'll give myself a 7/8 or 8/8 if I'm not too generous for this diagram. Maybe you should come in the comment section. What do you think would be my mark for this if you were the teacher for this diagram? Okay, moving on to the next one. I had to emphasize that because your diagrams are important and they are an integral part of your practical biology exams. Okay, so moving on to the next one now. The next one is going to say complete the following table on specimen K and M. So here you have specimen K, and then agricultural use, life cycle again, and seed leaf. You must know all those things. And then type of fruit again, the reason, and then the factors responsible for the condition of specimen N. If I can remember well, specimen N was your rotten tomato, right? So you see that's why they're asking for factors responsible for the condition. And then the next one is mention 3 methods of preservation of specimen M. So specimen M was your fresh tomato. So let's look at the answer flash as we look at that. Like I told you earlier, you can pause the video at any point and write them down so you can get these key points as you prepare for your exam. So you have the classification here: agricultural use, life cycle, and seed leaf. And then you have that fruits, fruit crop, vegetable, cereal, and all of that. And then the type of fruits for specimen M is a berry. Pericarp is soft and fleshy. And then factors responsible for conditions, you have bacterial infestation, fungal attack, poor storage, and attack by insect pests. And then you have methods of preservation. You have your freezing, drying, canning, and all of that. Okay, you have to take note of that. And then that's for that particular question. We are rounding up very quickly. We don't want to make this video very long. All right, so the next one here, we said state 4 observable differences. So take note of this. I think this is the first we are bringing for this. Observable differences between specimen M and N. They will ask you things like this, and they'll give you a table like this as well for you to put in your answer. So please take note of questions like this. They are very, very important. And they also ask you to mention 2 ways of preventing the condition of specimen N, how to prevent it. And they ask you again to draw a diagram of the transverse section of specimen M and label fully. Now take note, if you are asked to draw a transverse section, draw a transverse section. If you are asked to draw a longitudinal section, please draw a longitudinal section. So looking at the flash now, the answer flash to the questions there. So you see specimen M has a firm texture while N has a smooth texture, pleasant aroma, offensive smell. And so you can just read that up for yourself. Moving down, you see that the methods, 2 ways of preventing the condition of specimen N is by proper storage in a mesh bag, drying, and refrigeration. And then here again you have another diagram. So on this diagram now, you'd be able to tell, considering the things, the basic elements we've been mentioning so far: your title, your size, your clarity of lines, neatness of label, your details, your magnification. So don't forget to put this into consideration as you are practicing your diagram. A simple sketch diagram that would help you to do well as you prepare for your exam. All right, so moving on to the next question. You see here you are asked to state the name of the specimen and their functions for specimen P, Q, and R. And then coming down again, you are asked to state 4 simple steps on how specimen R is used. Specimen R should be the quadrat there. And then you came here again, we said state 4 biological processes affected by specimen P. And lastly, we said state 4 biological uses of specimen Q. So you need to pay attention to all those questions as we progress. Now let's quickly look at the answer flash to those questions. Okay, so looking at the answers, you see here that we had a summary. Specimen P is a thermometer used to measure your temperature. M is a meter, specimen Q is your meter rule used for length, and
Quadrat for estimation of population density. These are the steps in using a simple quadrat. Remember, I asked that you check out the video if you've not seen it before, so you can get more details on this. We're just trying to make this very short, so that's why we are trying to do this in a hurry. The 4 biological processes affected by specimen P are germination, fermentation, photosynthesis, and respiration. There could be more, but these are the ones we just covered in this video. Okay, so again, I had to bring 3 diagrams here. I brought 3 diagrams for you to draw, just to emphasize the need for drawing the diagrams very well. Okay, so let's see the answer flash to that specimen as we round up. Okay, so here you have the biological uses of specimen Q. It's P, so measuring plant height, that's a meter row, measuring seed length, measuring stem length, and then measuring leaf length. So basically, this is now the simple diagram for your quadrat specimen R, and then we label the wood frame and the string there. So that is how far we can go on the series. So we had to bring in this disclaimer. This disclaimer is just to tell you that these questions are not directly WAEC questions, but they were just simulated questions we had to put together. But we are very sure that practicing these questions will give you a smiling face at the end of the exam. So do well to put them down. Use these 24 hours effectively to study, understand, and then go over them one after the other, and you'll be fine at the end of the day. So if you've not subscribed to the channel at this point, please kindly do well to subscribe, like, share, and comment, that it will help to push this video to so many persons. You can also click on the notification bell so that you get our videos promptly as we upload them. Thank you very much for your support so far. It's been a wonderful experience, and I'm going to see you in the next one. Bye.